Good afternoon. My name is Elizabeth Snoddy. I'm from the University of Houston downtown, and we have been evaluating the efficacy of pine oil and lemon oil as biosafe alternatives to xylene in histological staining. So xylene is the standard agent used to de-wax and clear agent and clear tissues in the deparaffinization and clearing steps of standard histological staining. So, with that being said, why even replace xylene? Why would that even be important? That research question. Xylene has been the standard for over 100 years, so why even replace it? The reason is xylene is an environmental pollutant that's classified as hazardous waste. It's also a health hazard, and with repeated and prolonged exposures, it can affect the central nervous system, causing issues like nausea, dizziness, headaches. It's also a known teratogen and a suspected carcinogen. So it would be beneficial to find a biosafe alternative to xylene for the environment and for humankind. What large institutions like MD Anderson have done to mitigate risks to their employees is they purchased a multi-million-dollar robot, and that robot is the one that actually does the staining protocols. However, at small research facilities and even at the county hospital here in Houston, where finances are not abundant, all stains are still performed manually. So, can pine oil and lemon oil or lemon oil replace xylene in hematoxylin and hematoxylin is commonly referred to as H&E and Mason's trichrome staining. So in our methods, what we did is we first sectioned a formalin-fixed, paraffin-embedded tissues using a microtome at 7 to 10 microns. Then it was mounted on a slide. Then we did the deparaffinization step. Now the deparaffinization step, this is where we're going to replace the xylene with either pine oil or lemon oil. And in this step is where we basically just de-wax. We want to remove all the paraffin wax from the slide so that the stains can penetrate the tissue so we get good visualizations of the tissue structures later. Next, we perform the staining procedures, which is a series of ethanol, water, and stain washes. And lastly is the dehydration and clearing step. This is the step, again, the second step where we replaced xylene with either lemon oil or pine oil. And this step is basically, we're just removing any residual water, again, because we want to get good visualization of the tissue structures. Lastly, we imaged and blindly scored each one of the slides. They were scored on a scale of 1 to 3, 1 being poor, 2 being good, 3 being excellent, and evaluated 5 different traits. The traits were nuclear staining, cytoplasmic staining, uniformity of the slide, color balance, and clarity. Here we can see in our control group with the xylene at 40 time magnification, we have good color balance, we have good uniformity, we have excellent, I would say we have excellent uniformity on this one. We have good nuclear staining, good cytoplasmic staining. But when we take a step in and we go to the 400 magnification, we lose some of that clarity. So I would say that this is poor clarity because we cannot differentiate between the specific nuclei and see they all merge into one, looks like one cell. When we go to the pure pine oil, the experimental group, at 4E magnification, we don't have great uniformity across the slide, we don't have great clarity across the slide, the color balance is a little off, but at certain portions we have good nuclear staining, good cytoplasmic staining. But when we take that step in and we go to 400 magnification, we get excellent nuclear staining. We can differentiate between each of the nuclei within the mammary gland and the epidocytes. So across all of the experimental groups, we used mouse mammary tissue because we wanted to evaluate the same tissue type. So next, we want what we did is we wanted to quantify the data. So again, we had 5 histological research students grade each one of the slides, give them a 1, 2, or 3 number. After the data was aggregated, I performed a t-test, and the t-test said that there was no statistical significance, which means that we can conclude in our experiment that pine oil performed comparably to xylene in the H&E staining. So next we wanted to evaluate, would it do the same with Mason's trichrome? So we used pine oil and Mason's trichrome. We are in the midst of quantifying this data as we speak, but if we look at just the visualization between the control and the pure experimental group, well, they have very similar results visually. We just have not quantified this data, so this research is ongoing, but it is promising. So pine oil performed well. Let's try another aromatic oil. So we chose lemon oil. Again, this is preliminary results, and we still are running trials on the lemon oil. But from our preliminary experimental group, we can see that xylene and the control in the lemon oil performed comparably just by visual analysis. In conclusion, we can say that pine oil performed comparably to xylene, so it is a possible alternative to xylene in H&E staining, and possibly a possible alternative in Mason's trichrome. Lemon oil is a possible alternative, awaiting results. And the future work is we want to test other tissue types. Since we only tested mouse mammary tissue, we want to test lung, liver, kidneys, and see if it responds similarly. Next, we want to evaluate the longevity. Does xylene, pine oil, and lemon oil hold up the same way over time that xylene does? And lastly, we'd love to recruit a histologist or a pathologist to evaluate our slides quality. Thank you so much. I greatly appreciate you listening to my research. We have the acknowledgments and references.